Hello students, today in the present video we will study about the next topic, that is the seed. As you know, seed is a ripened ovule which contains the embryo or the miniature of plant body. The seeds of different plants vary in their size and shape. However, the general plan of structural organization remains almost the same. Next, that is the development of seed. As you have already studied in the previous video, the double fertilization in angiosperms initiates the formation of ovule into a seed. There are the number of changes usually occur in the ovule during the development of the seed. Number 1, the zygote develops into an embryo. Next, the triploid primary endosperm nucleus gives rise to a nutritive tissue called endosperm. The endosperm may persist or completely digested or utilized during the embryogenesis. The seeds having copious amount of endosperm tissue are called the albuminous seed. For example, wheat, corn, onion, etc. And the seeds in which the endosperm is used up are called X-albuminous. Example, bean, pea, gram, etc. The X-albuminous seeds usually store reserve food materials in the cotyledons. Next, the nucleus is generally used up during the development of embryo, but in some cases it remains outside the endosperm in the form of a thin layer, and that layer is termed perisperm. Next, the outer integument becomes hard and leathery testa or outer seed coat, which ensures survival of seeds. Next, the inner integument, if persist, forms the tegmen, Tegmen. Next, the micropyle remains in the form of a fine pore on the surface of seed. Funicle is transformed into stalk of the seed, and the helium marks the point of attachment to the stalk. Next, that is the parts of seed. Every seed has an outer covering called the seed coat. It develops from the integument of the ovule. The outer coat is called the testa and the inner layer is called the tegmen. The testa which is developed from the outer integument and the tegment which is developed from the inner integument. If only one covering is present in the seed, it is called the testa. The testa is hard, leathery, whereas the tegmen is thin and membranous. Sometimes tegmen remains fused with the testa. The outer surface of seed shows a scar or mark of attachment with the seed stalk. It is called helium. It is called helium. There is a small pore. You also see in this figure. There is a small pore called the micropyle, which represents the micropyle of the ovule. Some seeds also show the place of origin of seed coats, that is chelazal end or chelazal side, and the part of funiculus fused with seed wall, that is the raphe. The seed coat encloses an embryo which is differentiated into the radicel, which is differentiated into the radicel, plumule, cotyledons. The radicel, when elongates, gives rise the primary root, whereas the plumule gives rise to aerial shoot. As you seen in this figure, the number of cotyledons or seed leaves may be one, example in monocots, or two present in the dicots. The cotyledonary leaves are different than the foliage leaves. Sometimes they store reserve food materials, example gram, pea, almond, etc. Or sometimes they serve as photosynthetic organs in young seedling. The part of embryonic axis, the part of embryonic axis between the radicel and the point of attachment of cotyledons is called the hypocotyle. That is, you see here. Similarly, the part of embryonic axis between the plumule and the point of the attachment of cotyledons is called epicotyle. Next, that is the albuminous and ex-albuminous seeds, which is already described in the development of seeds. Next, that is the viability of seeds. Viability of seeds. Viability of seeds is defined as the ability of the seed to retain its power of germination for specific period of time. Some seeds lose their viability and fail to germinate after few weeks of their seeding, while some remain viable up to several years. The viability of the seed depends on number one, The conditions through which they pass after shedding. Number 1, the conditions through which they pass after shedding. Next, aging of the seeds. Next, stock of reserve food materials, etc. The viability is generally lost if the seeds are kept in dry atmosphere for the longer duration. The reasons for the loss of viability may be due to utilization or completely utilization of reserve food materials, denaturation of proteins and enzymes, and damage of embryo, etc. There are number of tests used to show the viability of the seeds or tested the viability of the seeds out. That is number 1, the germination test, and the another, that is the test with tetraxylome dye. Tetraxylome dye. Next, that is the structure of bean seed. Structure of bean seed, a dicot non-endospermic seed. A dicot non-endospermic seed, as you see in the figure. Bean seed is kidney shaped, that is, see here, having a concave side, that is this side, convex side, that is this side. The concave side is slightly darker and possesses a white elongated scar called helium. You see the helium, which marks the point of attachment of the seed with the funiculus. A small pore lies adjacent to the helium called the micropyle. Called the micropyle. The micropyle pore performs the function of absorbing water during the seed germination. There is a short ridge on the other side of the helium. It is called the raphe. The bean seed has two seed coats. The outer seed coat is called the testa. It is hard, thick, smooth, and variously colored. The testa, it develops from outer integument of the ovule. The inner coat is white, membranous, and thin. It is called tegmen. It is difficult to separate tegmen from the testa. The seed coats can be easily removed if the seeds are soaked in water for a few hours. Each seed encloses an embryo which can be examined after removing the seed coats. The bulk of embryo consists of two large, white and kidney-shaped cotyledons, that is this, or also called the seed leaves. The cotyledons store reserve food material in the form of the carbohydrates and proteins to provide the nourishment to embryonal axis at the time of seed germination and growth of young seedling. The cotyledons are attached laterally to the curved embryonal axis at the cotyledonary node, that is the node. The micropylar end of embryonal axis is rod shaped and slightly bulged. It is called the radicel or the embryonic root. The other side of the embryonal axis lies in between the two cotyledons. It is called the plumule or the shoot tip. The shoot tip remains protected by two small folded leaves as you seen here. The portion of embryo axis between the plumule and cotyledonary node is called the epicotyle, and the portion between the radicel and cotyledonary node is called the hypocotyle. Both the epicotyle and hypocotyle elongates rapidly when the bean seed or during the seed germination. Next, that is the structure of castor seed. Structure of castor seed, that is dicot endospermic seed. That is dicot endospermic seed. You see here, that is this. The castor seed is broader at one end and pointed, that is pointed at other end with elliptical outline. The color is mottled dark brown with hard, smooth and shining testa. Tegmen is thin, white and membranous. A soft, white, spongy structure called the caruncle is present at one end of the seed. The caruncle or aril is an outgrowth of micropyle. Almost in the middle of flat side of seed, a narrow like raphe is present. Almost in the middle of flat side of seed, a narrow ridge like raphe is present. There are two cotyledons which are thin, oval, papery and veined. Endosperm is fleshy in which store the reserve food material. Embedded in the endosperm embryo is present which contains an embryo axis with distinct radicel and plumule. Next, that is the structure of maize grain. That is an example of monocot endospermic seed. You see in the figure, that is this. Maize grain is in fact a dry, one-seeded, indehiscent fruit of Caryopsis type, in which the pericarp, means the fruit wall, is fused with the seed coat. The covering of the grain encloses parts. Encloses the two major parts. One is the endosperm and second is the embryo. As you see in the longitudinal section, that is this, the major portion of the grain is occupied by a large starchy endosperm. It is surrounded by a special one cell thick layer called the aileron layer. The cells of the aileron layer are filled with proteinaceous aileron grain, which plays an important role at the time of germination. The embryo consists of an upper shield shaped consists of an upper shield shield shaped scutellum, upper shield shaped scutellum, which is actually a reduced cotyledon. The scutellum is closely pressed against the endosperm and helps in the or transportation of nutrients from the endosperm to the growing embryo. At the time of germination and seedling growth. The epidermal tissue of scutellum, lying in direct contact with the endosperm, is both secretory and absorptive in function. It secretes certain hormones into the endosperm which synthesize enzymes these enzymes or newly synthesized enzymes responsible for the breakdown of reserve food materials into soluble forms. The soluble nutrients are then absorbed by the epithelial layer of scutellum and transferred to the growing embryo. The scutellum is attached laterally to the embryonal axis. One end of the embryonal axis points toward the pointed end of the grain. It is called the radicel. The radicel is covered by a protective sheath called the coleorhiza. The other end of the embryonal axis, which faces toward the broader end of the grain, is called the plumule. It consists of a shoot tip and few rudimentary leaves. The plumule is surrounded by a conical protective sheath called the coleoptile. Coleoptile. The coleoptile is capable of growth and covers the growing shoot tip till it passes through the soil during the germination. The coleoptile also possesses a terminal pore which allows future shoot to pass through it. Students, the topic of the seed is finished with this video, and the question on that may become up to 5 or 6 marks. Thanks.